Good morning to our beloved learners from all over the province of Romblon. I hope everyone is well and is successfully adjusting to the new normal of learning. Alright, so this morning class, we are going to discuss the most essential learning competency for week 1 in the subject General Chemistry 1. So that particular milk is describe the various separation techniques such as distillation and chromatography. For this milk, we have two specific learning objectives. The first one is describe what is distillation, and the second one is describe what is chromatography. So basically, we know that our beloved science teachers way back in our junior high school and even elementary have already had inputs in the different separation techniques. So to have a quick review, these separation techniques are different methods, actually composed of different methods on how to separate the two components of a solution, which are the solvent and the solute. Alright, but in this session, we will be discussing in particular the two separation technique, which is distillation, and the second one is chromatography. So class, what is this distillation for? So this technique, class, the distillation relies on the differences in boiling points of each individual component. When heated, the substances reach their boiling point and are converted to gas. Okay, so in our previous years, when we had our face-to-face classes, using distillation in an experiment in general chemistry is very common. So you have first to prepare your experimental setup. Of course, this involves the use of a couple of science equipments. To name a few, we have Bunsen burner, but in some cases, we already have our electric burner. Then we have the heat-proof mat, distilling flask, condenser, receiving flask, and thermometer. So as you can see here in the PowerPoint, this is an example of a distillation experimental setup. So for you class to have a clear grasp about this concept, let's watch this video about distillation. So we have many kinds of distillation. So in here we have steam distillation. So please observe the process class and some important points involved in the process. So this video is a very quick one, 1 minute and 46 seconds. Here we go.
How steam distillation works. Most essential oils are produced by steam distillation. Steam distillation was invented around 1000 AD by Persian alchemist and healer Avicenna. It has been practiced for thousands of years since then and is still one of the best ways to take freshly harvested or dried plants and extract pure essential oil. Take a look at this distilling apparatus. The bottom reservoir is filled with distilled water. Above is an alembic chamber that holds the plant material, often called biomass. The biomass is full of tiny sacs that contain essential oil. When the water boils, the steam passes up into the chamber. As the steam passes through the biomass, the temperature causes the tiny sacs to burst open, releasing the essential oil. The essential oil rises up with the steam, and together the oil and steam pass into a tube. This tube passes through a cold condensing chamber. The cold temperature changes the steam back into liquid, which drips out of the tube. The liquid collects in a new reservoir, where the water and oil separate naturally.
So
the water, which is called hydrosol, is left at the bottom, while the oil rises to the top. And lastly, the essential oil can now be collected and bottled.
Okay, so that's it. So in the previous video, in this video that we have played earlier, we have observed that the concept of distillation dated back 1000 AD, and primarily during the time of Avicenna. He was a physician and an alchemist during his time and must have been using this to extract essential oils to treat or cure ailments of pervasive diseases during his time. So as an application class of this process, separation technique, you can use this to extract different essential oils that can be used as fragrances and in most cases of medicinal value. I can still remember class when like 2 years ago, a year ago and 2 years ago, when we had our face-to-face classes wherein our students have performed this one. They have gathered the skin or the lemon peelings and extracted the lemon oil, which is called lemonine, and has utilized that in a certain research project that our students are having. So there you have it, class. That is basically what is distillation. Let's proceed to our next learning objective for our milk earlier, and that is to describe what is chromatography. So class, if you hear the word chromatography, it looks like it's a new word, right? And kind of gives you an impression that is this kind of difficult type of separation technique? But actually, this is a simple one and a very doable activity if you're gonna have an experimentation about this. So what is this chromatography about? Actually, class, chromatography as a separation technique has many kinds, all right? So we have more advanced form of chromatography process or experiments, but at this point, we will be tackling about paper chromatography. So this technique, class, relies on the concept that the solvent and the paper have different attraction to the components in a mixture. So, as I can still recall, a year ago, when we had our lesson in, I think that's general biology, when we are studying about pigments, in order for us to determine what are the different pigments of plants, so that covers particular vegetables and fruits as our samples, we have utilized, in order for us to determine what these pigments are, we should extract first those pigments and have it examined in a chromatography paper or a filter paper. So we have used a separation technique called chromatography. So basically, there you can see the integration of a concept across the curriculum, within and across the curriculum. Alright, so in addition, paper chromatography is a technique used to separate mixture of chemical substances into its individual compounds. So what we did in our experiment before is that we selected some vegetables, fruits, and flower petals, and then we have placed them in transparent glasses and named them according to what kind of vegetables, fruits, and flower petals they are. And then we measure 50 ml of alcohol in a measuring cylinder. So if you're doing this, you should handle the alcohol carefully. Then we pour that alcohol in each glass. Then we mix them well with the help of a steering rod. Then we let 30 minutes to pass. Let the solution be undisturbed for 30 minutes so that it can properly extract the juices from the cut vegetables and flower petals. Okay, and then you have to drain the alcohol with the help of a strainer. And finally, you will get the extract of all the samples. Consequently, class, you have to measure a strip of filter paper with the width of 2.5 centimeters and the length of 12 centimeters. So, as per activity sheet, it says that we have to use a chromatography paper, but we don't have that specific paper in our lab. So, with the aid of localization, we used filter paper instead. So you have to cut uniform sizes of 7 strips of filter paper. So because in our previous experiment, we have utilized 7 samples. So we also utilize 7 strips. Afterwards, you have to wrap one end of the filter paper around a barbecue stick and fix it with a paper tape or a tape. And all the others. And then this time, you have to make the filter paper stand in the glass containing alcohol. Make sure that the paper does not touch the sides of the glass, and one end of the filter paper must be dipped in alcohol. So let all the glasses be undisturbed for 30 minutes. After some time, you can observe that the solvent is rising up the water and is leaving some colored marks to the filter paper. So actually, those are already the pigments class in each particular plant samples of cut vegetables and flower petals. Then you have to take out or remove the filter paper from each glasses of extract and let the paper dry. Okay, then when it is dried already, it's time for you to perform your analysis. You have to observe the different pigments present in each of the filter paper with each specific sample therein. So as you can see here, class, in the PowerPoint presentation, this is an example of the findings of our student last year. So here you can see the different colors. So you have their bright yellow, dull, bright green, dull green. Okay, so those different colors have specific names. Those are actually the pigments. So for the blue-green, that's chlorophyll A. For the yellow-green, that's chlorophyll B. For the bright yellow, that's carotene. For the pale yellow-green, that's xanthophyll. And for other samples, wherein the color revealed from orange to red, violet to blue, those are actually the pigment anthocyanins. Right? So in that particular example or experimentation, we have actualized this separation technique called chromatography. So there you have it, class. We have already described what are these two separation techniques, namely distillation and chromatography. I hope you learned something, and thank you so much for your time listening. Thank you.